Hello everyone and welcome once again to Biolympian. Today's video is going to be a practical demonstration of how to dissect a flower, and the flower we are selecting today is of the family Malvaceae. It's Hibiscus rosa-sinensis, or a China rose flower. And before we begin, I would like to show you some of my dissection equipment. Right, so the first tool we are going to be using today is a scalpel. As you can see here, it's a bladed scalpel. It's not the blade is not inbuilt. I have to attach the blade separately to this to the holder, and the blade is very sharp. So if you're doing this experiment at home or in a laboratory, you should be very careful about this point, right? Because it's a sterile surgical blade, and this is the same blade that's used for surgeries, right? So it has to be really sharp. Okay, and we need to utilize the sharpness for making the cross section of the ovary later on. That's why I've chosen such a sharp blade, right? Okay, so the next tool that we are going to be using is a pair of tweezers, or forceps. We call them forceps in scientific language, not tweezers. And this is the small one, this is the larger one, right? I'll just scoot them over a bit, and both have different purposes. And finally the needle, right? The needle, this is for pointing out different parts of the flower, and also for making tiny incisions that will help us separate the parts of the flower. And I will also need a small piece of cloth. This is for removing the slime that usually is present at the bottom of the whorls of the flower, like the calyx or the corolla. And since it gets in the way of our experiment quite a bit, we need to remove the slime, right? So this cloth is for that, right? And we'll also need to prepare a slide at the end of the experiment showing the cross section of the ovary. So I have a slide right here. This is my slide. You see, it's been used before, that's why it's not crystal clear, but that's all right. This is the slide, and of course when we have a slide, we need a cover slip to cover it, right? And so this is my cover slip, right? This is my cover slip. Okay, so these are all the equipment that we will be requiring for the experiment today. You can also have a watch glass full of a little bit of water if you just want to wash out the slime a bit better, but it's not really necessary, or to be more precise, essential to the experiment, because you can just wipe it away. You can just wipe away the slime from your fingers using that cloth, right? Okay, so I think we may begin the experiment now. This is the flower that we are going to be dissecting today, and before dissection, it's always a good idea to inspect the flower for a little while, because then we can glean quite a few facts about the flower, quite a few of the details of the flower. And of course, this is a Hibiscus rosa-sinensis flower, or a China rose flower. And if you know about morphology of flowering plants a bit, then I am sure you can describe the familial characteristics of this flower. It belongs to the family Malvaceae, and one of the characteristic features they have is the presence of the epicalyx, right? I'm going to be taking my needle, and let's point out where the epicalyx is. You see, this is the epicalyx. This is one of the let's say leafy bits of the epicalyx, and it's called an episepal actually, right? This leafy bit, it's called an episepal. Okay, and you see it's all around, right? It's all around. You see there's another green leafy bit above, and there's one green leafy bit below, right? So the part below this thing right here, this is an episepal, and it's a member of the whorl called the epicalyx, right? And if I just take this episepal as a reference, and I put my thumb right here, and this is the first episepal, so this one will be the second episepal, this will be the third episepal, this will be the fourth episepal, and finally this will be the fifth episepal, right? So there are 5 episepals in total. So we say that the epicalyx is pentamerous, right? Pentamerous as in 5-membered whorl. Okay, you'll see as we progress throughout the video, you will see that the entirety of the flower is actually pentamerous. All the whorls of the flower are actually pentamerous, right? Okay, now that we have talked about the epicalyx, let's now talk about the calyx. Okay, you see this is the calyx above the epicalyx. This part above the epicalyx is called the calyx, and on close inspection you will also find 1, 2, 3, 4 and 5, right? So there are 5 sepals in the calyx. The green leafy bit that's present in the calyx, each one of them is called a sepal, right? And if you have studied a bit about the morphology of flowering plants, I'm sure you can appreciate the fact that the sepals are joined together, and so we call this configuration as gamosepalous, right? We call this configuration as gamosepalous. Okay, so all the sepals are joined together, they have a common base, and their apices are different. Their apices are separate, right? So we call this configuration as gamosepalous. Okay, now let's move on to the third whorl, the colorful whorl. What we notice first, we don't notice the green leafy bits down there, right? Because that's just drab and boring. It's just like a leaf, any other leaf, right? But this red bit, this is why we pluck these flowers. This is why we use these flowers in our rituals. So this red bit right here, these 5 members of this whorl, these are called petals, right? I'm sure you know about the nomenclature. These are called petals, and the whorl as a whole is known as the corolla. Some of the pollen is getting stuck in my hands, but that's all right. This entire whorl is called the corolla, right? The corolla. And when we remove the calyx, you will be able to appreciate the fact a bit better that these petals are all attached separately. These are not adherent to each other. That's why we call this configuration as polypetalous, right? Polypetalous. The petals, since they are separate from each other, they are not joined to a common base, we call the configuration, the arrangement, as polypetalous, right? Now this is quite a small flower, so I'm not sure if you can see the inside of it, but this red tube you see emerging out of the base of the flower, the proximal end of the flower. This red tube is actually the staminal tube, and since there's only one of these tubes, we call the configuration of the stamens as monadelphous, right? Monadelphous, that is united into one single staminal tube, right? And you see these tiny black and yellow bits right here that you can I think you can see on the video. These tiny black and yellow bits, these are the stamens, right? These are the stamens. And if I just highlight one of the stamens right here, you see this is the filament where my needle is currently. This is the filament, and the tip of it, the distal end of the stamen, we call it the distal end of the stamen. It is the anther, right? And the anther is where the pollen grains are produced in the hibiscus flower, right? And so you can also see a few kind of feathery little bits here. You see? Do you see? These are these kind of hairy, kind of feathery little bits here. These are called the stigmas, right? These are called the stigmas. So what can you infer about the flower, considering the fact that both stamens and carpels, since stigma is a part of the female reproductive whorl of the flower, so what can you glean about the flower? It's bisexual, right? It's bisexual. It's a bisexual flower. It's also known as a hermaphrodite flower, right? So these are the stigmas, and beneath the staminal tube is the other part of the carpel, or the female reproductive part, the gynoecium, the style, right? And when we cut into the flower, when we dissect the flower, we'll observe the ovary, which is present at the base, protected by all these whorls, because the ovary is the most The ovary is actually where the ovules are housed, right? So it has to be protected. And a couple more facts: the ovary is hypogynous or superior, means it is present at a level that is above the base of the other floral whorls here, so we call it superior. And the aestivation of the calyx, as you can see, the aestivation of the calyx, since the ends are all just touching each other, right? These ends are all just touching each other. We call this aestivation as valvate. And in case of the corolla, since see, one margin is above the adjacent petal and one margin, the other margin, see, it's not visible, it's below the adjacent petal, right? See, if you observe this petal right here, this one, you see one margin is above the petal that's adjacent to it on the right hand side, and the other margin, it's not visible here because it is below the right margin of the left adjacent petal, right? So we call this kind of aestivation as twisted. If you observe, all the petals have the same kind of configuration, right? All the petals have the same kind of configuration, all the 5 petals, and we call this kind of aestivation as twisted, right? Okay, so that's about the basics of the flower, and now I think we can get down to dissecting the flower, right? Okay, the first step to dissecting this flower is to take your scalpel. This is my scalpel, and just make a small incision. Just make a small incision along the calyx right here, right? Be very careful not to damage the whorls underneath, especially not the ovary, right? You see, now that I've made the incision, the two adjacent sepals have been separated, right? The two adjacent sepals have been separated. Now hold the flower like this using the corolla and just go around incising the bases of the episepals, right? Of the episepals, and just go around. Be very careful not to damage the ovary, because the ovary is the most important bit. I think it's better if we actually separate the episepals, right? We don't need to display the episepals in our experiment, so we can just separate them one by one. You see, I have to expend quite a lot of, I have to separate them quite forcefully, right? So it's quite adherent to the base of the calyx, the epicalyx. Now I can just make an incision around the base of the calyx like this without damaging the ovary underneath. Okay, with very delicate hands, right? With great care, taking great care not to damage what's below, right? Okay, I think we have incised the base well enough, and now I can take my tweezers and just lodge them underneath one of the calyces right there. I can lodge them underneath one of the calyces right there, and then I can just separate the entire calyx as a whole. Once I've got a little bit of it out, I can just use my fingers and I can there's a little bit of adherence right here, so I'm using my forceps to just incise that bit, and now I can separate the rest of the calyx as a whole, right? You see, I have separated the calyx as a whole, and you can see that it's pentamerous. It's got 5 sepals. You see, 1, 2, 3, 4, 5, right? It's got 5 sepals. It's pentamerous, and this is the calyx. I'm putting it aside for now. We are going to be needing it later when we display it as a whole. Okay, so that's the calyx separated. Now let's separate the other whorls, namely the corolla, right? So the corolla, in case of the corolla, since it's a polypetalous corolla, right? So I can just remove the petals one by one. You may need to make a few incisions using the scalpel. Just make a tiny incision like this, and actually you don't need to make any incisions. You just tear out the petals one by one, right? This is the first petal, this is the second petal. You see it's not adherent at the base. It's all the petals are separate, right? You tear out the third petal. Be very careful not to damage the ovary. Tear out the fourth petal. This is the fourth petal. And finally the fifth petal, right? The fifth petal. So I've separated the 5 petals, and now you see I can arrange them like this. I can arrange them kind of like this, and these are my petals, right? So I'll keep this aside as well for now. We're going to need it later. But for now, let's proceed to the next bit, the important bit, which is the separating the staminal tube and the carpel, right? So the carpel, actually, in dissection, you can just bluntly dissect the staminal tube, right? You don't need to show it whole in maybe a practical examination or a practical scenario when you're doing this in the laboratory, right? But I want to show the staminal tube, so what I'll do is I'll separate the ovary from the bottom, and you see I've dissected the style, right? I've separated the style. Usually you have to separate the gynoecium hole, but since I want to show the staminal tube today, I'm going to bisect the style, and the remainder actually can be pulled out from the top like this. Okay, the remainder can be pulled out from the top like this, and actually that got dissected in the middle once more, but that's no matter. And now I have this staminal tube as well separated. You can see there are 5 stigmas. So this is a pentacarpellary syncarpous gynoecium. There are 5 stigmas if you can count, and the intervening style and the ovary at the base that is still attached to the thalamus, right? That is still attached to the thalamus, and this is the staminal tube. This is the staminal tube. You see it's hollow in the center. You can see it from this way as well. It's hollow in the center. Actually, we got the entirety of the style then, since it's completely hollow. You see, there's nothing inside, right? So this is the staminal tube, and these are the stamens that are adherent to the top of the staminal tube, and this is the base that surrounds the ovary, right? The base of the staminal tube surrounds the ovary. Okay, and we call this type of configuration as infinite stamens, because you see there are a large number of stamens and it's impossible to count them all, right? So we call this, when we represent it in a floral formula or a floral diagram, we on a floral formula actually, we call it as infinite stamens, right? So I can separate these whorls as well. Okay, so I think that's the entirety of the flower dissected. And now we take a white piece of paper, and we display the whorls that we have separated, right? So first, of course, is the calyx. We display the calyx like this, right up top. We display the calyx like this, right up top. Actually, it's better if I do it like this. Yeah. So the five sepals of the calyx. Then we display the corolla. This is the corolla. The 5 petals of the corolla. Alright. This is the androecium, the staminal tube. Usually we don't need to display the staminal tube, but I wanted to show it to you today. This is the staminal tube. This is the ovary with a bit of the style attached. And the 5 stigmas which are adhered together. And now we just use the big forceps to kind of change the configuration so it looks a bit better. Alright, so these are all the whorls of the flower separately dissected. You see, it was a neat dissection, and we can appreciate all the separate parts of the flower quite clearly in this dissection. And that's the dissection part over. But now we have to make a slide of the ovarian cross section, right? We have to make a cross section of the ovary, and we have to make a slide out of that, right? So to make a cross section of the ovary, I'll first take the ovary and I'll keep the rest of the whorls separately. I'll just take the paper like this and I'll keep it aside. Now take the scalpel. This is a very delicate procedure, and just separate the top cleanly. You can either use a scalpel or a blade. Okay, and it has to be a very thin section, right? Very thin section, so we can appreciate all the parts very clearly. The first section is almost always not great. The first section is not great. This one is a good section. It's very thin. See, you see the diameter is really thin, and this one I can actually put on my slide. I have my slide right here. Okay, I have my slide right here, and I can actually just transfer it to my slide using the needle. It's a bit slimy. That's why it's adhering to the scalpel, but no matter, we can surely get it. Yeah, so I've put it on my slide, and now let me just attach the cover slip. I'll attach a cover slip after putting a drop of water and draining the excess water with a brush or the corner of a piece of blotting paper or a filter paper, and I put the cover slip over it. You don't need to use any stain. You can use a saffronin stain if you want, but it's not essential. I'm doing this one without the stain, and you see this is the prepared slide, right? You see there's no excess of water because I've drained it all, right? All the water is inside the cover slip, and this is how you prepare the slide. Okay, and finally let's put this slide under the microscope and see what it looks like, right? So yeah, you should be seeing the picture of the slide on your screen right about now, and I think you can appreciate the 5 chambers in the ovary, the 5 locules in the ovary. We call the chambers as locules, and it's got 5 chambers because it's actually a pentacarpellary gynoecium, that is syncarpus. So the 5 different carpels, ontogenetically, the 5 different carpels have been fused together during development, right? So you can see the ovules as well within the ovary, and within the separate locules. There's 2 ovules per locule in this particular case. And lastly, you need to talk about the placentation, which in this case is axile because it's along the axis, because the septum is present along the axis of the ovary, right? So I can take off the slide now, and this was the end. This is the end of our discussion today, of our practical demonstration today on how to dissect a flower belonging to the family Malvaceae. And I hope you liked the video. See you later. Thank you.